It's on a day like this that I think, why am I wearing a tie and a sport coat? It's the middle of summer, but here I am. I have a closet full of ties and sport coats, so I've got to wear them somewhere. My name is Paul Beck, and I'm the chair of the steering committee of the Emeritus Academy. And I'm really pleased to welcome you to the last of our lecture series. And we've had 8 talks this year, including today's. We have put together a really interesting slate of talks for next year and are kind of working out the sequencing of those. And you'll hear in a couple of weeks from us what the features are for the 2018-2019 year. I want to take this opportunity to thank the Office of Academic Affairs for sponsoring this lecture series, and especially to Kay Wolf, who is the Vice Provost for Academic Affairs, and to Melissa Kemp, who are the ones who kind of guide us through all of this. And we are very, very grateful to them for their help. I also want to thank the College of Arts and Sciences Digital Media Studio for videotaping all of the 2017-2018 lectures. The videos will be posted, or the video from this one will be posted on our website probably in a couple of weeks. All the others are there as well. And if for the final exam you want to review what went on during these particular lectures, I invite you to take a look at the website. Let me also mention that there'll be a short business meeting after this lecture, and at that meeting what I would like to do is introduce the new members of the Emeritus Academy. And then finally, I'm always reminded to remind you to leave your name tags on the table outside when you leave so that we can use them again. It cuts down, obviously, on our costs. Well, it's my pleasure to introduce today's speaker, Jack Wood. Jack is a professor emeritus of physiology and cell biology in the College of Medicine, has spent over 30 years at Ohio State University. His research has focused on enteric nervous system, in particular neurophysiological, biophysical, and neuropharmacological investigations. I wasn't sure I could get all that out, of what he was the first to call the brain in the gut. This work, especially his seminal 1981 article in the Annual Review of Physiology, established and nurtured this specialty of neurogastroenterology. He also may be, and this you probably don't know, the most famous alumnus of Commerce High School in Oklahoma since Mickey Mantle. And most people in this room know who Mickey Mantle was. Today he will talk to us about our second brain, the brain that is in the gut. Jack?
Well, thank you, Paul. Like Paul with his jacket, I'll just take the hat off. It's not a pretty sight, but I'll tell you, I can go any place in the world, from Beijing to Cairo to wherever, and someone will say, wearing one of these hats that always say Ohio State, and someone will say OH, invariably. Doesn't matter where. Let me. There we go. Well, I'm happy to have the opportunity to talk to you all about the brain in the gut, the little brain in the gut. I named it that. And I think in the physiological reviews a long time ago. This is the gut. This is the gut, and this is what the medical students see in my earliest lecture in the gastroenterology block. We talk about all the specialized compartments that are lined up along the gut. They learn all about those, and then they also learn about the sphincters separating each of the specialized compartments, and that they learn that the compartments don't like the stuff to come back into them from the back. So the function of the sphincters is to prevent this reflux or back movement of material from one compartment to the other. Well, like I say, I always show that slide. That's the introductory slide to the gastroenterology block and also to the physiology course. So one of the students sent me this postcard. I don't know how long it had been. We'd been into the course, but they sent, I wasn't going to show you that until later. So anyway, they said, you know, Dr. Wood, we thought you would be the one to enjoy this slide. That's what we talked about. And then actually there was another statement at the bottom of the slide, or of the postcard, and that was this message here. So the little brain in the gut, as Paul mentioned, I called it that way back in 1981, just based on all the work we were doing with recording from neurons, looking at neurotransmission in the enteric nervous system, and then comparing that with gut behavior. And it was clear that this was an independent, integrative nervous system, analogous to what makes a cockroach operate, or what makes a fly fly, or crayfish work. Same sort of concept of an independent, integrative nervous system that works, in our case, virtually below the stomach, independent of the big brain. You see what Mother Nature done. You have 100 million neurons in your enteric nervous system, all wired up into microcircuits, very much like the spinal cord and the brain. This is an image of the enteric nervous system, both parts of it, in a newborn gut, so you can see all of it there. You see what Mother Nature, or evolution, or Buddha, or God, or whoever you believe is responsible for the way we are. What they've done is evolving the gut, and from a very simple organism. I first did some work on the squid, which is very much like our enteric nervous system, but rather than having long connections between the big brain and the little brain and sending messages over unreliable pathways, what it's done is established integrative circuitry in an integrative nervous system right next to the effector systems that require control. Now, those effector systems are systems like the musculature there, like the secretory glands, and like the blood vessels that all require control and coordination of their behavior. The enteric nervous system, the little brain, here it is in red. This is a segment of gut. There is the mucosa and the two muscle layers. This one is the longitudinal muscle coat, and this is the circular muscle coat. And the two of them work together and are innervated by what we call the myenteric plexus of the enteric nervous system between the longitudinal and circular, and the submucosal between the circular and the mucosa. And we always point out that all of these components are very much like any other integrative nervous system, like our big brain. For example, they have a program library of different kinds of behaviors that the little brain controls. There's also feedback control, negative feedback control. The physiologists know this is required for set point control, keeping different things constant like blood pressure, reflexes, and processing kind of information. One of the things we did a long time ago that demonstrates the independent nature of the enteric nervous system. If we take a colon from the guinea pig, place it in physiological solution, and then place an artificial fecal pellet. There's an innovative, creative graduate student that decided to put lubricant on a fecal pellet and see what would happen if she pushed it into the upper end of a piece of colon. Push it in here, and then what you find, now no connections to the body at all, in physiological solution, propels the fecal pellet slowly to the opposite end, and then ejects it at the opposite end. So no connection to another nervous system, no connection to endocrine system, no connection to any other part of the body. We use in earlier years, just during these studies, we use a drug called tetrodotoxin. Some of you who know the pufferfish in Japan know this is a very potent toxin, but it's a very useful tool for us because it selectively blocks neurons because of the ion channels they use compared to the muscle. So you can observe the physiology of the muscle without the enteric nervous system. And so that's what we've done here. But you see, now we place the fecal pellet in the colon. And notice it doesn't move, but look carefully, and you'll see that the musculature now along here, especially the circular muscle coat, is not silent, but now is continuously contracting. And this is the nature of the musculature, is that it's autogenic. It's very much like my colleagues there that study cardiac muscle. They're all electrically coupled, and it's autogenic. So this requires a special kind of control that we call with neurons that keep it inhibited and control it mainly with the inhibition of the musculature. Well, we know now, as all my years, 40 years or 45 years of studying the enteric nervous system, we know that, and we've learned that if there's anything not working right in the little brain, there will be symptoms. The individual will have symptoms that they will relate to their gut or to their digestive tract. This example in Hirschsprung's disease also is evidence for us that the gut can't work without the little brain, without the enteric nervous system. This is a little 13-year-old boy that doesn't receive a lot of medical attention, but this big tummy, this big belly, is all due to impacted feces in his colon, in his large intestine. This is not the same person, but here's a large intestine, colon, and constricted in a segment, terminal segment of that colon from a relatively newborn. But the hallmark of Hirschsprung's disease is that we call it a megacolon, an enlarged colon with impacted fecal material. And then with the distal segment, some length of the distal segment of the colon being constricted, and not only being constricted, but also contracting all the time, like fibrillating heart muscle. Used to be thought that the deficiency, the problem, was in the megacolon. And it was studied for years and years and years until we came along. But now we know that terminal segment has no enteric nervous system. It's congenital, the birth is without it. But the reason we tumble to autogenic aspect of the muscle, or you see it here, is it's continuously contracted because especially the inhibitory innervation of the enteric nervous system is missing. When you can find an animal model for a disease, you're well ahead of the curve in studying the disease. And way back in the 1970s, when I was at the University of Kansas Medical Center, came upon a mutation in which the little mice are piebald, that is, they're black and white. But it turned out that the piebald mouse, the little white one there, with the genetic mutation in the endothelin B receptor, also has Hirschsprung's disease, also has megacolon, normal sibling. And they're easy to pick them out because the little guys, the Hirschsprung's disease, are white, so you identify them very early on. Here are three large intestines from a normal mouse, that black one. Here's the one from the little guy, the white one with megacolon and the constricted terminal segment. And this is just oftentimes with these mice, much as is the case for children, newborns as well, they will be born with an imperforate anus. The anus doesn't open. So I will use this to demonstrate the differences between Hirschsprung's disease and this case of imperforate anus. But more than that, demonstrate the difference in that segment when there's no enteric nervous system there as opposed to the normal mouse. And you see what happens is the little mouse with the imperforate anus, which hadn't opened, has a megacolon. But in this region, if we stain for the little brain, this all is innervated. Another thing I'd like to talk about is the idea that the enteric nervous system stores a library of programs for different behaviors of our gastrointestinal tracts. And I've shown 1, 2, 3, 4, 5 programs that are stored in the little brain, in the gut. This one here, I call it, or we call it, postprandial program, which is a mixing motility program. After a meal, this is kind of the mixing behavior that's occurring, and that's programmed by the enteric nervous system. Another of the programs is one where there's no motility whatsoever. The gut's silent. We call this ileus or physiological ileus as opposed to pathological ileus. Another one here is the interdigestive program, or we call it the migrating motor complex. This one, when you switch, as soon as the meal's over, digestion is complete, absorption is complete. Now the system switches from the postprandial program to this interdigestive program, and it starts in the stomach with big contractions of the stomach, and that's when the stomach empties large particles. It won't empty anything bigger than a millimeter or two until this interdigestive phase. And then interdigestive phase, when you watch it with contrast material in the lumen, you see a propulsive contraction starts and goes down so far and stops. Then it'll start a little further and go even further and even further until it gets to the end, and then it starts over. What this is doing is clearing out debris, clearing out undigestible material, but also the gallbladder contracts when that starts, because you see, you don't want bile sitting around too long in the gallbladder, because this will precipitate into gallstones if it becomes too concentrated. So this is what we call the interdigestive program. Another one is, you've seen pictures such as this probably of the colon of the large intestine of humans. You see it's divided into little sacs like this with muscle contraction in between the sac. This is squeezing the feces. The idea is this is squeezing water out of the species, because much water is absorbed from the large intestine. But just another of the programs that are stored in the library of the little brain in the gut. Another one I'll mention a little later is called power propulsion. In this case, power propulsion is a very powerful propulsive contraction that moves very rapidly through the intestine, small and large intestine. This example is in the colon, and you see within less than 30 seconds or so, it started way up here, big, enormous contraction, and propagates all the way to the end. Now, I'm going to tell you in a few minutes that this is really, when you have the crampy lower abdominal pain, when you know you probably need to get to the bathroom, this is the motility, that's the program that's running in your large intestine. It evokes the pain. We'll get to that a little bit later. And another of the programs is labeled here as the emesis program. In this case, instead of the little brain programming for propulsion in the aboral direction toward the anus, propulsion reverses, starts in the middle of the small intestine and then propels contents very rapidly back toward the stomach. So this is emetic. So you get the idea of what I'm talking about in terms of programs. This is a very nice example. This was given to me by Professor Hans Ehrlein in Germany, outside of Stuttgart. But Professor Ehrlein was a veterinarian. And Professor Ehrlein was studying the movement of the digestive tract with the animal swallows barium, and then under the fluoroscope, imaging the movement of the barium inside the segment of gut. In this case, the dog had had a meal, and what is running is the postprandial program. The postprandial mixing program is running. See, here's a constriction, here's an open compartment, constriction. But it turned out that the dog, with the meal, had also had rat poison in it. So the owner of the dog had brought the dog to Professor Ehrlein for treatment. Professor Arline treated it much the way we would do in the ER with a little kid that we thought had ingested poison. You invoke emesis, give them. And in this case, you will see it was apomorphine was used to evoke emesis. So what you see in this video is the mixing movements of the postprandial program, and then the very sudden conversion to the emetic program and the power propulsion of moving the stuff not in the aboral direction, but toward the stomach. So this is the motility program after a meal, when there's a meal in the stomach. And then you'll see the conversion. You'll see it someplace here where the program changes, right about here, I think, right in here somewhere. Okay, here goes now the emesis program. Here's the power propulsion. Visualize that, because we'll talk about that also as it occurs in the distal, in the colon, the distal small intestine. Very rapidly propels the contents from the mid small intestine into the stomach, and then the dog retches. So an example, the idea of a program library in our digestive tract. I talked to medical students about this, and I don't know, it's been just a couple or 3 years ago, this bright medical student said, "Hey, Dr. Wood, you know what you're talking about there are apps. You're talking about essentially cell phone apps. You touch one of them and a program runs." And this is the analogy. So I now use the apps analogy instead of programs analogy. Well, among those apps, I want to mention just one that we call the defense app, because the defensive app is designed to get threatening materials, threats out of the lumen of the digestive tracts, large and small intestine, also stomach. It's emesis that does it in the stomach. But to get threats out of very quickly out of the lumen, infections, food allergens, that sort of thing. And the big point to make about that, and you think about it from the standpoint of your own experience, the point to make about that is the defensive app. The sequence of events as the app begins to run is, first of all, there's massive secretion. Lots of secretion into the lumen. This volume accumulates. And then this is followed by power propulsion going in the aboral direction, much like what you saw going to the stomach in that earlier video. And then the internal anal sphincter opens, and this is when the individual experiences the urgency. Knows he'd better get to the restroom because there's something bad's can possibly happen. Well, there is a program. There is a defensive program that's designed to eliminate threats from the lumen. One of the ways this program gets called up in the functional gut is by some cells that are located. You see, here's the lining of the gut. This is the mucosa. There's several kinds of cells in the mucosa, but under the lining, under the mucosa, are these cells. We call them mast cells. Now, what I'm going to tell you about mast cells is mast cells signal the little brain when there's something threatening in the lumen. When mast cells detect a threat in the lumen, they then release mediators, a signal to the little brain that starts the defensive app. Here are mast cells. This is on an electron micrograph, all enlarged. But the thing about these is mast cells have about, in those granules in there, have about 35 or 40 chemical mediators that get released in different situations. For example, it's these cells that release the mediators that call inflammatory cells into the lumen of the gut in the initiation of inflammation. Look, see, there's an inflammatory cell, and there's one. You see they're nestled right up to this mast cell. And the mast cells They're pathologic in many cases. Here's an example. Here's an example of a friend of mine, Gilbert Castro, down at the University of Texas in Houston. See, this is a normal histological section of the small intestine. See, there's a mast cell stained there and another mast cell. Now, Gil infected the animal with an intestinal parasite, in this case, Trichinella spiralis. You'll remember Trichinella was in the polar bears when Admiral Byrd was up there, because part of their life cycle is the what do you call, the second generation or third generation, live in the muscle, and people ate the muscle and they got trichinosis. But after infection with the intestinal parasite, you see a big increase in the numbers of mast cells and the mastocytosis. We also know that in the intestinal tract of irritable bowel syndrome, that in irritable bowel syndrome, there's also a mastocytosis. Here's a normal colon mucosal biopsy, and 3.3 or so mast cells, as the red ones here, per unit area. And this is a biopsy from an individual with diarrhea predominant irritable bowel syndrome. So the major point I want to make here is when the defensive app, when the defensive app runs, it's designed to, first of all, initiate secretion, large amounts of secretion into the lumen, followed by propulsive motility to eliminate that, and the sensation of urgency. My point here is that when the defensive app runs, that there are these symptoms. First of all, we've all experienced some abdominal malaise. Something's not quite right. It's gurgling down there. Cramping abdominal pain. The cramping pain that oftentimes will make you do this is power propulsion occurring and moving those large volumes of secretion toward the anus. And then the fecal urgency, because we have two sphincters, actually. We have the internal sphincter at the end of the colon, and then also an external sphincter. When that internal sphincter automatically relaxes during the propulsive motility, now then the only protection is that external sphincter, and it's under control of the spinal cord. It's not under the control of the little brain. So these symptoms that most all of us have experienced at one time or the other, these are common symptoms, for example, in postprandial irritable bowel syndrome. We just mentioned irritable bowel syndrome. There are also symptoms associated with food allergies, nut allergies, all the allergies you can think of that we've had, that folks have, and have symptoms associated with them. These are the symptoms. In enteritis, for example, infectious enteritis, those E. coli that are being sold to us in romaine lettuce, those symptoms are all very much the same. In radiation-induced enteritis during the days of radiation treatment for cancer, same symptoms. In ulcerative colitis, if you know someone with ulcerative colitis, the very same symptoms. Cramping abdominal pain, acute watery diarrhea, and urgency. Microscopic colitis, Crohn's disease, another inflammatory bowel disease, collagenous colitis. But the last one there is stress. And this has always been interesting to me my granddad, some of his terminology is pretty colorful. I can't really want to repeat it here, but for folks that get stressed or what happens to someone that gets stressed. Maybe in a few years, if I'm still around, we have an entire presentation just on stress and the physiology of stress and the effects of stress on the digestive tract, on the gastrointestinal tract. But all of these that most everyone in this room recognize, reflect the running of that the defensive app. So what I'm telling you what I'm telling you is here's the little brain. Here's the little brain of the gut. Incorporated in the microcircuitry is this program library. And one of those apps in that library is the defensive program. And what mast cells do is they release mediators. And when the little brain sees one of those mediators, then it says there's something that's caused a mast cell to release that mediator. So we better let's run the program to get the stuff out of the lumen of the bowel. So the little brain, the little brain also has several kinds of neurons, just like our spinal cord. The neurons that innervate the musculature, that controls all those movements we looked at for the musculature. Neurons that innervate the secretory glands. I'll mention those as being secretomotor neurons in the little brain. And then also another of the effector systems that gets controlled are blood vessels. For example, when there's massive secretion, like as the defensive program runs, the secretory glands are activated to secrete water, sodium chloride, and bicarbonate. Now then, the other thing, those neurons then innervate the blood vessels, and they at the same time will dilate the blood vessels, so there's increased blood flow to support the secretion. And that's another example of just how smart Mother Nature was in wiring up the enteric nervous system. And then, of course, the behavior, the control of the musculature, the glands, and the blood vessels get integrated into overall behavior, just like my big brain's integrating my movements of this and my failure to press the right buttons on this clicker here. But then coordinate all of that into intestinal behavior. And I mentioned before, it includes hypersecretion and power propulsion. But then these have symptoms. These are associated with symptoms. Neat little extremity here if you got something bad in the lumen of the gut. But we have to deal with the symptoms, and the symptoms are cramping, abdominal pain when power propulsion's moving stuff downstream. The secretion, massive secretion, which is the acute watery diarrhea. And of course, being afraid that the central nervous system is not going to keep the external sphincter closed is the fear of a accident to the laundry. I thought I would just show you one example. I mentioned food, commonality of symptoms, but for example, in food allergies, here's the guinea pig. We did quite a bit of work with guinea pigs in this respect. What we've done with, I'm going to tell you about with the guinea pigs, is we'll have them drink cow's milk for 3 weeks or so, and then replace the milk with tap water after 1 or 2 days, and then we can take a segment of gut out, put it in the organ bath, and record from the neurons with microelectrodes. And then the control for this are guinea pigs that have never been exposed to milk protein. So just one example is what we've done here is microelectrodes in a neuron in the wall in the preparation, and now we add to the bathing solution beta-lactoglobulin, which is just milk protein. And what happens when we add that to the organ bath is, you see, this is the membrane potential of the neuron, and it increases in this direction. That tells us that the neuron's becoming more excitable, and then it begins to fire, and these upstrokes are firing of the neuron. Here, the same guinea pig had never been exposed to egg protein, to ovalbumin, and that's a control, and that doesn't do it. But if we fed the guinea pig ovalbumin instead of milk, ovalbumin would then trigger this response. And like we hope good scientists, then it's quantitative, it's dose, it's concentration dependent, the concentration of the allergen. In this case, what is happening is to an allergen, the immune system makes antibodies. Most everyone here, I think, are aware of this already. Doesn't matter if it's shrimp, tree nuts, peanuts, whatever. The immune system has made some antibodies to the nut or the shrimp or whatever, or some component of it. And those antibodies, they're in the circulation, and they circulate around to the gut and the mast cells in the gut. And those antibodies attach to the mast cells like this. These little Y's here are antibodies. For the guinea pig, these are antibodies that recognize milk protein, because the animal's been sensitized to the milk protein. Now then we add to the bathing solution the antigen and milk protein again. Now what that does is, because the antibodies recognize it, the antigen reaches the milk protein, reaches the antibodies on the mast cells, cross-link those, and that degranulates the mast cells and releases the mediator that the enteric nervous system is going to see and run the defensive program. So let me finish just to give a little so you can understand the diarrhea that associated as a common symptom. I mentioned that the enteric nervous system, the little brain, has neurons that we call secretomotor neurons, because they innervate the secretory glands, those that are going to secrete sodium chloride and water. Every time this neuron fires, it releases a neurotransmitter that evokes some secretion. So it's easy enough to see that anything that increases the frequency of firing, there's going to be more secretion. So in this case, the mast cells, and we've shown this experimentally, mast cells, one of the major mediators is histamine. Histamine binds the receptors that include the secretomotor neurons, and this is what stimulates the secretion. And then this is what we describe as neurogenic secretion, or neurogenic diarrhea, as opposed to osmotic diarrhea, that's agents like laxatives, like sodium, milk, and magnesia, and that sort of thing. So this is the way when the defensive app runs, this is the way you get a short story on how you get the secretion that leads to the acute watery diarrhea. Another example of that I mentioned to you several times, because I want everyone to think when the little kid comes to you and says, God, my belly hurt, and he bends over, you'll know that this is power propulsion occurring in his colon. And the way we came to appreciate that was this kind of a study. This is a guy I got to know well up at the Medical College of Wisconsin. But he used dogs, and he sensitized dogs to parasites, to intestinal parasites, and in this case, again, Trichinella spiralis. And so then he puts some sensors, attaches some sensors inside the gut wall or in the lumen, so that every contraction of the circular muscle can be recorded at this spot, this spot, this spot, and this spot. Now, in this case, in this particular example, the dog was sensitized to the parasite, Trichinella spiralis. So then ground up some worms and injected it into the dog, had a little catheter in there, and immediately that evoked this very strong contraction here. And then within time, here is in minutes, you see about, less than a half a minute, started here and run all the way very quickly toward the anus. When this, I watched them do these studies up in Milwaukee. When this is happening, when you see this on the recording, the old dog sometimes is screaming. He's not really screaming, but he's twitching. He could tell that he's really in some lower abdominal, having some kind of crampy pain. And I won't go on to tell you how the technicians that also have a bucket behind the dog, how they don't particularly like this experiment. So let me finish by first acknowledging what's made me successful. And I have a feeling, well, certainly my colleagues will agree, that what really makes us successful as scientists, especially physiologists, are all the people that have worked with us in our laboratories over the years. And this is, I've tried to list everyone that's been worked on one or the other of my projects on the little brain, the enteric nervous system in the gut. Also need to acknowledge the National Institutes of Health. This is National Institutes of Diabetes and Digestive and Kidney Diseases. They funded me funded my research function of the enteric nervous system continuously from 1971 to 2016. Non-fundable impact and a percentile score from the clinical, investigative, and molecular gastroenterological study section terminated the work on the enteric nervous system at The Ohio State University in 2017 because they didn't fund my grant finally after all those years. So let me say this is the end, and thank you very much for listening to some pretty
recognize people for questions.
Okay, sure. Paul told me I could recognize people for questions. Let me turn off this mic that's making all the noise when. Okay.
Jack,
Yeah.
you made some comment about immunological memory in the enteric nervous system and the gut, and with a specific focus on gluten hypersensitivity.
On gluten hypersensitivity. I don't know about gluten hypersensitivity, but I know the way it works is it evokes the commonality, the same symptoms we've discussed, right, Rolf? And so that must mean that the immune system is somehow sensitized by, or however it gets sensitized. And I'm not so sure we understand how lots of sensitization occurs, but this means gluten, the immune system has learned to recognize that and know it, and then the antibodies are there, and then those antibodies will find their way to mast cells, bind to mast cells, and next time gluten comes along and cross-linked, that then degranulates the mast cell, and then the little brain sees the mast cell mediators coming, and that says, run the program, get that stuff out of there. But in more detail, Rolf, I don't know. There's probably something about the way the immune system recognizes gluten, and I don't know. Yes, sir.
Yes, I have a question about connections between the higher nervous system and the gut nervous system. Can you talk yourself into getting diarrhea?
Yeah.
I mean, is it possible that cerebral cortical can work through the autonomic system to say, I really hate it here. I think I'm going to have diarrhea. The big brain can. We used to call this operant conditioning. That people, for example, I remember having some students at the University of Kansas who could put a pressure sensor between the fingers, and they could will it to change. I don't know so much. I don't know examples, but I've heard a story one time about in India, a Maharaji or something like that. This is pretty gross, but you know what I do is gross. This is all about beetles. Go ahead.
I'm not a good person to go to dinner with. But, but you know, the idea we could sit on a tub of water and aspirate water by willing on how it.
That's a yoga trick.
That would really be
I think Rolf Barth would like to know how to do that. Huh?
Ralph, any?
Speak for yourself.
No,
the reason I asked is it's a long and involved story, but I'm trapped up in a tree. I was out bow hunting, and my tree stand fell apart, and I'm trapped in a tree. And the only way I could get down is to hold and shinny, and that wasn't working real good. I spent 20 minutes up there sweating bullets. I finally just had to grab it and slide down the tree, which I did, hit the ground, and immediately had your propulsive diarrhea, anxiety, whatever. So I'm wondering if my brain said, when you're done with this, we got something else to take care of.
that's the stress aspects of it.
Okay.
That reminds me, I'll tell the story very quickly, but I was lecturing in Egypt, and a guy said to me, I was on a safari in Africa, and what you say I believe entirely, because we were on this trip, and we'd run over with the Land Rover a snake, big old snake, and so the driver had put that snake in a paper bag that they'd had for lunch or whatever, put it in there, and put it up on the hood of the Land Rover. Now they go a little further, and they come upon this troop of baboons. And the alpha male, the baboon, he jumps up on the hood and starts flipping the windshield wipers and banging on the windshield. And And he looks down and he spotted that bag, that paper bag. And he grabbed the paper bag up, opened it up, and then there was just what you described when you hit the ground.
Okay.
But I could tell you, there is, the control is a little different for the stomach because the
Okay.
stomach requires a little more complicated to control the stomach.
Okay.
So it does involve the big brain.
Okay.
Sometime
All right,
we'll
well,
talk about it.
I'd love to talk to you about it.
Anybody else? Yes, sir.
Yeah, I've been hearing recently about something called leaky gut syndrome. Does that have anything to do with?
Sure it does, because those cells that I showed you in the mucosa that are lined up like columns, like so, they're connected. They have little junctions between them, and that keeps a lot of bad stuff from getting across, so the mast cells would pick it up. But stress, for example, we've done quite a lot of work on that. Those junctions, they'll open up a little bit during stress. So the possibility for something that, the lumen of the gut is pretty dirty, and for that to get across, now then mast cells pick this up, and that's what will trigger.
This is a layperson's question.
Well-
Do you have an opinion on probiotics, which are now so highly praised? And
Well, let me say that most probiotics are bacteria, aren't they?
Yeah.
And probiotics have to pass through the stomach. And the stomach has acid that's about, pH is about 1 point something or 2 point something. So I don't think many bacteria make it to the positive use of probiotics, in my view, where they really do work, is something called Clostridium difficile colitis. And everybody I play poker with 6 other guys. When I started talking to them about fecal transplants, these guys really didn't know what to make of that. But it seems it's very effective so that's an example, but it's not the example you're asking about. Is like Lactobacillus
right, for
and
the
things
general
like
population.
that.
Yeah.
So, I've always heard as a kid that the Dutch that ingested a lot of yogurt and Lactobacillus didn't have, So, in my view, that I think the story's still out, but I don't know.
Interesting.
Yes, sir. So, Paul.
Other questions? Well, again, Jack, thank you very much. I always thought things were going on.
Hold it, one more question.
Oh, yeah.
It's a kind of sentimental question. You had that very disturbing slide of the 13-year-old boy.
Mm-hmm.
Could he be cured?
Absolutely. Absolutely. If he'd had adequate medical care, what the surgeons do is they resect that constricted segment where the little brain is missing, and then pull it back, and there are 2 or 3 surgical techniques for doing that. I had a friend named Holzschneider, one of my years in Munich. It was a pediatric surgeon, and he would do those surgeries in the little kids, and reconnect the colon to the anus, and then he would always study them. He'd bring them in once a year, put the balloons up to see if they had the common reflex of you distend it and the sphincter relaxes. And he always swore to me that the enteric nervous system adapted to that and behaved like an adult enteric nervous system over time after the surgery. So that's the only comment I can make about that. Except,
Glad to hear.
Pretty good comment. Yeah.
Okay, other questions? Well, Jack will be here, and if you have further questions, come up and talk to him. Let me now turn to the business part, which will be very short. Thank you, Jack. Is this on? Yeah, I think it is. First, I want to take this opportunity to thank the 3 departing members of the steering committee who have served us very well for the last 3 years, giving us valuable service. Lois Rossau, who is here, Rolf Barth, who is over there, and Terry Miller, who is right here. I should add that special recognition, I think, is owed to Terry. Terry was a founding member of the Emeritus Academy, and I think it's fair to say there would be no Emeritus Academy but for his efforts and we are very grateful to him for that. Thank you, Terry. There'll be new steering committee members for 2018-2019. They will be Artie Nelson, Russ Pitzer, and Jack Rawl. Mary Jo Fresh will be the new chair, and the chair-elect is Joe Donnermeyer. So there's kind of a changing cast of characters here. If you're interested in being on the steering committee in the future, please volunteer. That's where we get our members. Now let me turn to the new members of the Emeritus Academy. And what I'd like to do is introduce them one by one. Honors come with certificates, and I have certificates for each one of them. I apologize in advance that I may mangle some of the names, and I would like to be corrected if I do. Let me start with Hojat Adeli. See here? Please come on up.
Thank you.
Congratulations.
Thank you. Thank you very much.
Oh, sure. Hojat is in the Civil, Environmental, and Geodetic Engineering Department in the College of Engineering. Did I get that right?
Okay, yes, I'm affiliated with other departments also.
Okay, in the medical school, right?
Just for
Yes,
neurology,
that's right.
neuroscience, and biomedical.
Okay.
Informatics. Actually, that's where my research focus is right now, in the neuroscience and neurology part of the campus.
It's probably worth saying that if you use rollerblades, you could easily get back and forth between the College of Engineering and Medicine. But you probably, if you're like me, you don't. Thank you very much. Secondly, Will Clark. Is Will here? Okay. TK Daniel. TK, come on up. He was disguising himself in his application as Philip, but I think I know this guy. Congratulations.
Thank you very much, Paul.
There you go.
Thank you.
And he is from the Department of Educational Studies in the College of Education. Next is Christopher Ellison. And Christopher is from the college, or the Department of Surgery in the College of Medicine.
Right. Thank you very much. I
Congratulations.
look forward to it. Appreciate it.
Next is Ralph von Fries, and he is from the School of Earth Sciences in the College of it's actually the College of Arts and Sciences.
Thank you.
Congratulations. Oops, wrong one. I'm glad I checked. There you go.
Yep.
And next, I'd like to introduce Sabra Weber. She is from Near Eastern Languages and Cultures in the College of Arts and Sciences. And Comparative Studies. Congratulations.
Very much.
And there are some other members who are not, other new members who I believe are not here today. So let me just read their names. If they are here, please come up and I will give you a certificate. Paul Casamassimo, Joshua Dressler from the College of Law. Casamassimo is from Pediatric Dentistry in the College of Dentistry. Herbert Ackerman from Animal Sciences in the College of Food, Agricultural, and Environmental Sciences. Rajiv Shivpurti in Integrated Systems Engineering from the College of Engineering. And Joseph Zeiden from Near East Languages and Cultures in the College of Arts and Sciences. Again, all of them, I think, deserve our congratulations, and a hand would be